flower structure. Okay. So again, for ovaries and ovules, if you use the example of a pea plant, the ovary is all of the outside wall of the pea, and then each of the individual peas and their surrounding layers of leaf-like tissue is an ovule. Okay, let's see. Okay, so now we're ready to talk about the female side of the equation here and what happens in the female part of the plant. And this will all take place in the megasporangium. So within the carpel, there'll be structures set aside for cells to undergo meiosis. And once again, now we have a megaspore mother cell instead of a microspore mother cell. It's diploid. That cell will go through meiosis, but we're not making gametes. We're only making megaspores. So at the end of meiosis, we'll have 4 meiotic products, and here they are. And in contrast to the microspores, which are 4 equally sized cells, now we're going to have 1 big cell that got a lot of the cytoplasm, and 3 little microspores that hardly get any. So you can guess what happens. These 3 little guys degenerate and never become an egg, and the 1 big guy sticks around, and that will become the structure that gives rise to the egg and other cells. So these are called microspores, and they're going to have to go through, again, mitosis to become multicellular and then make the egg down the line. So 1 surviving megaspore here. So that, remember when we talked about oogenesis, it was the same kind of thing where there was a mechanism to make sure that one of those meiotic products got the majority of the cytoplasm. And that was useful in the life history of the organism because that cytoplasm could contain a lot of starter products to jumpstart the metabolism of the zygote once it was fertilized. Okay, so this megaspore, it's haploid, and now it's going to go through the business of making the multicellular female gametophyte. And it does it in kind of an odd way. What it does is it's first going to cause the nucleus of that megaspore to replicate through cytokinesis, to replicate, but we're not going to get cytokinesis. So the nuclei will be replicating, and they're going to be here in a common cytoplasm. So there'll be 8 nuclei, and then the plant says, oh, well, wait, we forgot to do cytokinesis, so let's catch up. And then once we have these 8 nuclei, now cytokinesis will occur and divide that up into cells. Only instead of 8 cells, it only makes 7. So one of the cells has 2 nuclei. Okay, so here are the 7 cells then, that is the result of these mitotic events. And that whole structure is called the embryo sac, and that is the female gametophyte. So then what are these 7 cells? One of them will become the egg. So that's a haploid cell, it will become the egg, it'll be fertilized by the sperm, by 1 sperm. Next to that are 2 cells called synergids, and as we mentioned, their function is to secrete a chemoattractant molecule to draw the pollen tubes close to the micropyle, which is the opening here between the surrounding leafy tissues or integument's. Then up at the opposite side of the embryo sac, we have 3 cells known as antipodal cells, and apparently the function of these cells is kind of mysterious. If they have any function, we don't really know what it is, and we just observe that these cells will degenerate shortly after their formation. And then that leaves 1 central cell that has 2 nuclei, and those are known as the polar nuclei, and that big blue cell is called the central cell. And that will be a cell that also combines with 1 of the sperm to make endosperm. I think we get to, okay, I'm going to come back to that point, but what has happened here is the pollen tube has gone down and grown, and we have 2 sperm that are on their way here. And so plants actually have a double fertilization event that happens. And that's why we need 2 sperm. one of the sperm will combine with the egg. So you have a haploid sperm and a haploid egg joining together and establishing the plant zygote. Then that zygote goes through mitosis. The second sperm is going to join up with these two polar nuclei in the central cell and now make a triploid cell. And that triploid cell will also go through mitosis and make the endosperm. So it's just kind of a different way of doing things relative to animals. We have an embryo that gets made, but then we also have a triploid endosperm tissue that's sort of like the extra embryonic tissue in mammals. And its job is to provide nutrition and support for the early embryo and keep it alive and healthy, but those endosperm cells themselves do not become part of the new individual. Okay. So here's just another slide where I've diagrammed that out. Double fertilization. And I think we already talked about that as well. The synergids will eventually degenerate, the antipodal cells degenerate, the central cell becomes the polyploid endosperm and the triploid endosperm. The egg cell gets fertilized and becomes the embryo. And then these leafy structures here known as teguments, they're going to wrap around this newly fertilized embryo, and this structure will become the seed. So we have in here a diploid cell plus a triploid endosperm surrounded by teguments, which is the seed coat. And then outside of that was the ovary. The ovary is the part that becomes the fruit. So here's some examples of this. A coconut, for example, think of the hard outer shell. That came from the ovary structure. Then what's labeled here in A, this is a relatively small multicellular portion, that was the embryo. B is these flaky cells, that is the coconut meat. Those are endosperm. And then these cells actually generate kind of a large vacuole in the middle filled with some fluid that's coconut milk, and that's also basically endosperm. So if you've had a pina colada, you've drunk endosperm. So here's the seed and the fruit. So the walls of the ovary could sometimes just form a protective structure, but in other cases it's actually the walls of the ovary that proliferate as the bulky mass of the fruit. So say an apple is going to have the seed itself is composed, the dark black parts were the tegument. Inside of the seed you'll see endosperm in the egg. And then all that fleshy mass of the apple came from the walls of the ovary. Okay, any questions on this part so far? Okay, let's go ahead and take our break, and then I'll come back and talk a little bit about one particular example, plant patterning within the leaf. So it's 8:42 now, so let's come back.